Hello, good day, Homo sapiens. Welcome back to studying Prescott Microbiology. Today's topic is Archaea Cell Wall, and Archaea Cell Wall is just like bacteria. No, that's not correct. Archaea Cell Walls. First of all, what are archaeas? Archaeas are the prokaryotic organisms, or you can say microorganisms, that were first confused with the bacteria because they resemble quite mostly with the bacteria, but as soon as the study is going, they started figuring out that they were quite different from the bacteria. First, they live in extreme environment. Okay, we are talking about archaeas, okay, before going to the cell walls of archaeas, for those who don't know what archaeas are. So they first were confused with the bacteria, and after their ribosomal RNA studies, their cell wall studies, and their habitat studies, they found out that these are quite different from the bacteria, even their characteristics and how they react to stimulus and stuff like that. So they were classified as a distinct domain in the prokaryotic cell. So now we have bacteria and archaea, two different domains in the, no sorry, not domain, two kingdoms. Can you stop? So about their cell wall characteristics, they are more similar to gram positive than gram negative. And the tests, the gram staining test, they either give the gram positive characteristics or gram negative characteristics in their test, like they might be purple or they might be pink, but when you study their structure, their pink color isn't that of the cell wall of the gram negative bacteria. So the gram staining doesn't work on archaeal cell walls because the test comes false. Like even if we see that the test is gram positive, actually their cell walls are not gram positive. So there are three main things that are different in archaeal cell walls than the bacterial cell walls. The first thing is they have peptidoglycan, but not the peptidoglycan of the bacteria. As you can see here, we have, where was it written here? We have N-acetylmuramic acid, NAM and NAG. We have to, guys, before coming to archaeal cell walls, you should go towards the bacterial cell walls, because if you don't know what the bacterial cell walls look like, I don't think you will be understanding what the archaeal cell wall is like as well, because I have already explained everything in the bacterial cell wall. So then this N-acetylmuramic acid, NAM, which we call in short, we have NAM and NAG as a glucose or polysaccharide in the bacterial cell wall, right? But here we have, instead of this N-acetylmuramic acid, N-acetyl aminuronic acid. That's what we have different. By the way, NAM and NAG are the basic building units in the peptidoglycan in the bacterial cell wall. Like they are repeating structures, they are repeating units that come over and over and over again. So if you change the unit even a little bit, of course the whole cell wall is going to be different. And because of this changing in the structure, we have another characteristic that is different from the bacterial cell walls, and that is the bonding structure. In bacterial cell wall, we have beta 1 to 4 glycosidic bonds, while in archaeal cell we have beta 1 to 3 glycosidic bonds. Now you guys will be thinking, what is the difference between glycosidic 1 bond and 2 bond? So here it is. You know we have carbon numbers, right? So first, do you know what carbon number 1 is, carbon number 2 is, carbon number 3 is? So when we start counting the carbon, we started from one side, right? And that one is known as carbon 1. We do have categories at what side is the carbon number 1. Like you can give this one the carbon number 1 as well. But no, we have a definition to either is this side carbon number 1 or either is this side carbon number 1. I forgot which one it was, but the starting one carbon is the carbon number 1. Now we have another polysaccharide. Remember, in polysaccharides, these are carbohydrates, so we have carbon backbone in them, right? So we have carbon numbers given to the C or whatever we have given to them. So in this polysaccharide, in this carbohydrate unit, we have carbon 1 to 4, right? And now think about this another carbohydrate unit, and think about it also as a 4 carbon. I mean, it doesn't necessarily have to be, but just for the sake of understanding. Now, if the bond is between carbon number 1 and carbon number 3, we call that as beta 1 to 3 bond, glycosidic. Glycosidic bonds are formed between two glucose units, not glucose unit, the saccharide unit, the carbohydrate units. We call that glycosidic bond. So if it is between carbon 1 and to carbon number 3, it is 1 to 3 glycosidic bond. And if it is between carbon 1 and carbon 4, then it becomes the 1 to 4 glycosidic bond, beta 1 to 4 glycosidic bond. This is necessary because bacterial cells have this beta 1 to 4 glycosidic bond. Usually there are very rare cases in which we have 1 to 3 glycosidic bond in the bacteria. Archaea and bacteria are different in the cell wall because bacteria form 1 to 4 glycosidic bond because it is more stable, it has more energy conservation, and it is less focused on the structure. It is more common and more energy focused, while glycosidic 1 to 3 bond is more focused on the variation in structure. It has more branches, it has more ability to bond more because it came to the carbon number 3 bonding. Because of their structure, because of their bonding, that the structures get diverse in the archaeal cell walls. Bacterial cell walls are less diverse than as compared to the archaeal cell walls. If you see the bacterial cell wall of gram-positive, all of the gram-positive will be somehow similar, right? But if you look at the archaeal cell walls, they will be all somehow different from one another, and you will be thinking like, is this even archaea or not, or is this some other thing, right? That's how much they are different from one another. So the first one was that instead of simple NAM and acetylmuramic acid, we have an acetyl aminuronic acid, right? The second thing is we had beta 1 to 3 glycosidic bond instead of beta 1 to 4 glycosidic bond, okay? The third thing is we talked about the amino acids that connect these NAM and NAG to one another. Do you see these? These are the amino acid 1, 2, 3, which we had 4 in the bacterial cell walls, and if you look at here, it's also 4. The bracket ones can be present and cannot be present. I mean, only in archaea, right? But it should be only this alanine and gluconic acid. No, sorry, not gluconic acid, glutamate. So it should only be these two, but it's more like an option. It is not always present. So excluding these two, these 4 and these 3 are always present in archaea and in bacteria. Usually in bacteria we have what do you call that? DAP, short form, or DAM, whatever. Instead of that we have lysine, just as in some bacteria we had lysine as well. So if we had lysine, it will have a interbridge, but I think the archaeal bonding between these amino acids are different. Like, look at this. In bacterial cell we had the bonding between the last amino acid, like here, here. The last amino acid, and it was linked with the third amino acid, and it was cross-bridging, it was cross-linking, because it wasn't end-to-end, it was one at the end and one in the middle, and the same case is happening within the archaea. Now you will be asking, what is the third thing that is different in archaeal cell walls than the bacterial cell walls? And that is these amino acids. We had D amino acids, dextrorotatory amino acids in the bacterial cell walls, while we have L amino acids in these archaeal cell walls. Bacterial cell wall has, first of all, let me tell you what is D and what is L. This is only the isomers of a same type. Like if we have glutamate and one form is, wait, let me again bring out this pencil. Alright. So we have, you can say carbon, carbon. Let's say we have O at this side and OH on this side, right? And now if you rotate it, if you make a mirror isomer of that, and it will be like this, OH, C, and O, right? Now you see they are mirrored to one another, but if you put this thing above and put it above this, it won't be overlapping at one another. Even though they are mirrorly overlapping, if you take it like this and just overlap it and this way, right, it might overlap, but if you pick this up and place it directly above it, it won't be overlapping it. So they are mirror images of one another. Now there is one that refracts the light in the right direction, that is the dextrorotatory, and there is one that refracts the light in the L direction, left direction, that is levorotatory, right? That's how their names were given. Now the L one is more common. L amino acids are more common and are present in proteins. They are mostly present in amine. So the peptidase is the enzyme that degrades the proteins that has amino acids, right? They are mostly prepared for only L types of amines. But bacteria, they want resistant cell walls, right? So they have D amino acids in their cell wall instead of L amino acids. So the peptidases that degrades the protein that has L amino acids, those peptidases cannot target the D alanines, right? They can't do it. But in archaeal cell wall, they have L amino acids instead of D. So now you will be thinking that the bacteria so now the bacteria are more resistant because they have D alanines instead of L alanine. But no, the archaea, even though has L alanines, they are more resistant than the bacterial cell walls. And you will know that because the archaeas can survive in extreme temperatures, extreme cold and extreme hot. Now, like there is an example given, the thermobacterium. I think there is a direct example of it. Yeah, thermoproteus and the sulfolobus here. So these types of archaeas have the cell walls that are resistant, and bacteria can survive in high temperatures. So it's not only because of their plasma membranes, it's also because of their cell walls. Now their cell walls not only have these structures, right? These three that we mentioned, the bondings, the L amino acids, and the different form of NAM, right? The cell wall resembles to that of the gram-positive. We did say that, right? And usually their tests are also gram-positive. Now if you look at this, look how much thick is this, 30 to 40 nanometer. The gram-positive thickness was maximized at the 70 nanometers. The maximum was 70 nanometer, while in the RQ1 it is 30 to 40 nanometers. Even though the thickness is not that, but it is heteropolysaccharide, right? In the gram-positive one, it was homopolysaccharide. Means the NAM and NAG was repeating itself, and it was quite homo, means there were not that much different polysaccharides in them. But in case of RQ1, they have different types of polysaccharide, and they even have proteins in them, which the bacterial cell walls lack. They only have proteins as channels or as signals or as receptors, but other than that, they don't contribute to the structure of the cell wall. But in archaea, the proteins do contribute to the structure of the cell wall, and sometimes they are actually a lot in the quantity than that of the, what do you call it? They call it if in that case, in gram-negative, we had lipopolysaccharide, in this case, we have heteropolysaccharide, right? There is no lipid in them, but the polysaccharides are in diverse amount, quantity, or quality. So this is just the general structure. Now the archaeas usually are like this, but there are archaeas that are like this as well. There is a word that is written to describe it, and that is chondroitin connective tissues. Wait, where was it? Yeah, polysaccharide complex polysaccharide, that is the chondroitin sulfate of the animal connective tissues. Chondroitin sulfate of the animal tissue. Now chondroitin sulfate, you will be wondering, this is the carbohydrate that is made from the repeating unit of the disaccharides, right? Disaccharides, and they are connected to one another by 1 to 4 glycosidic bond and 1 to 3 glycosidic bond. Means 1 to 3 glycosidic bond and 1 to 4 glycosidic bonds both are present in them, and so the structure becomes even more complex. And because these are disaccharides, they are repeating themselves in a complex manner, so they are more like that, the heteropolysaccharide. And when you add the protein, it becomes even more complex. So they are different from the bacterial cell walls. These were the three main things that were different. Oh, okay, I was talking about the structure. So usually they look like this, but in some cases when there is highly dense negative charge on them, right? And there is, because of that charge, negative charge of them, because of that another layer is formed above that, and because of that the structure becomes somewhat like this, these circular layers, as you can see. See, this is how they protect themselves even more, double layer. And I think that outer layer sheet that is formed, I think that is proteins. They are proteins. Now, so either it will look like this or either it will look like this. And I think this case is more present in the extreme temperature, extreme temperatures. Sorry, that was this distraction. I will just read it because I think that were the main points. And regarding this diagram, I think this is what I explained more in the bacterial cell walls. So if you want to go dig deep into the diagram, you can see the bacterial one, because they are not that much different, but they are different. Before they were distinguished as unique domain of life, the archaea were characterized as being either gram-positive or gram-negative, because they were confused with the bacteria. They were acting like bacteria, but they weren't actually bacteria. However, their staining reaction does not correlate as reliably with a particular cell wall structure as does the gram reaction of the bacteria. Like if in bacteria there is a gram staining becomes gram-positive, that means the bacteria is gram-positive and the structure will automatically be peptidoglycan layer, right? But here, if the bacteria even becomes the gram positive, if the archaea does react as gram positive, and if you look into the structure, it won't be peptidoglycan. It is pseudo peptidoglycan, and other than that, there may be sheets on it like that, those circular structure. So even it's more like a false result. Like you will have to see the structure to understand if the gram staining was correct or not. So archaeal wall structure and the chemistry differs from those bacteria. Yeah, the structure and the chemistry are different. Archaeal cell walls lack peptidoglycan and can exhibit considerable variety in terms of their chemical makeup. Okay, they do lack peptidoglycan, but you can say they lack true peptidoglycan, even though they have pseudo peptidoglycan, or you can say that peptidoglycan is also called as murine, right? So they have pseudomurine structure. Some of the major features of the archaeal cell wall are described in this section. Okay. Wait. What am I doing? Stop it. Yeah, many archaea have wall with single thick homogeneous layer resembling that in the gram-positive bacteria. So they are saying that the archaeas that are living in moderate conditions, not the extreme conditions. So they are more likely gram-positive. They are more like the gram-positive ones. These archaeas often stain gram-positive. Their wall chemistry varies from species to species but usually consists of complex heteropolysaccharides. Alright, so with the homogeneous layer, they do have heteropolysaccharides. See, now they are contradicting themselves. First they are saying that they are homogeneous, then they are saying that it has heteropolysaccharides. Because the gram-positive bacteria had peptidoglycan. Oh, okay, so the peptidoglycan, they lack true peptidoglycan layer. So they do have NAM and NAG that were repeating, but with that, even that, heteropolysaccharides are also present. So it's a mixture of kind of gram-positive and gram-negative, excluding the lipid material that is present in the gram-negative. For example, methanobacterium and some other methane-generating archaea, methanogens, have wall containing pseudomurine. Pseudomurine. Okay, so all of them don't have murine, but some of them have pseudomurine, a peptidoglycan-like polymer that has L amino acids instead of D amino acids in its cross-links. N-acetyl I can never say this name right. Aminouronic acid instead of N-acetyl uramic acid, and the beta 1,2,3 glycosidic bond instead of 1,2,4 glycosidic bonds. Okay, so even in archaea, they all don't have the 1,2,3 glycosidic bonds. Some have 1,2,3 glycosidic bonds, not all of them. So I was a little bit wrong in that regarding that. So mostly are like gram-positive with no differences, just heterolipopolysaccharides in them. And some bacteria have these kind of things. Like the bonding is different, the NAM has that different. Other archaea such as Methanosarcina and the salt-loving Halococcus contain complex polysaccharides similar to chondroitin sulfate of animal connective tissues. Right, they are saying that they are not just simple polysaccharides, like NAM and NAG are simple polysaccharides. It can be more complex than that. By the way, you can see the chondroitin sulfate structure, and so you will understand. Many archaea that stain gram-negative have a layer of glycoprotein or protein outside their plasma membrane. Okay, so the ones that act like gram-positive, they have heteropolysaccharide plus the homogeneous layer, right? While the one that act as gram-negative, their cell membrane has a plasma membrane. Outer to the plasma membrane, they have glycoproteins layer of that. The layer may be thick as 20 to 40 nanometers. Sometimes there are two layers, an electron dense layer and a sheet surrounding it. Some methanogens, I feel like I have said everything wrong. Oh my gosh. They all are talking about many archaeas, some archaeas, these archaeas, all archaeas. So they are saying many archaeas that stain gram-negative. They are not talking about all the archaeas, they are only talking about the archaeas that stain the gram-negative, and even in that many of them. And their layer can be 26 to 2040 nanometer, while maybe the gram-positive archaeas may have even more thicker layer than that. Sometimes there are two layers, an electron dense layer and sheet surrounding it. Some methanogens, Methanolobus, salt-loving archaea, Halobacterium, an extreme thermophile, Sulfolobus, Thermoproteus, and Pyrodictium have glycoproteins in their cell wall. So mostly the gram-negative ones are living in extreme conditions. In contrast, other methanogens, Methanococcus, Microbium, and the Methanogenum, and the extreme thermophile, D. Sulfococcus, have protein walls. So some have glycoproteins in their wall and some have a protein wall. Like some have pure protein wall while some have just glycoproteins here and they are not pure protein wall. So that was that. Looks like I was quite confused in this one because I confused a lot of facts in that. So as simple as that, there are two types of archaeas. One is gram-positive, one is gram-negative. Even though the reaction can be false at times, like you have to see the structure for you to consider it gram-positive, gram-negative. It's not like bacteria. So the gram-positive ones that come out as gram-positive have walls that have that don't have peptidoglycan, but they have a homogeneous thick layer, a single homogeneous thick layer, just as in gram-positive, and they have complex heteropolysaccharides as well. So the gram-positive one may be not the gram-positive ones. Yeah, those that stain gram-positive have a wall thick homogeneous layer and may have heteropolysaccharides, while the one that come out as gram-negative mostly come out as the gram-negative. They have an extra layer that is glycoprotein or protein, right? And that is 20 to 40 nanometers thick. And they, of course, if they are protein, so they are electron dense layer, and there will be sheet surrounding that. So in general, it was that as that. And in the gram-positive ones, there are some that even though they don't have peptidoglycan, there is a pseudomurine-like structure, like means pseudo-peptidoglycan structure, and in that they have these N-acetyltelos-aminuronic, oh my gosh, instead of N-acetylmuramic acid, and they have 1,2,3 glycosidic bond instead of 1,2,4, and yeah, and they have L amino acid instead of D. So I see, so this is just the small portion of archaeas that have these L amino acid and these characteristics, so all of them are different. Or should I make another video? How do I put a disclaimer that the starting is a bit confusing to people? I mean, if someone reached till the end of the video, they will know that the start was a bit way too vague and not detailed. So yeah, it's not my fault if someone watched half through video, okay? So this is just the structure of pseudomurine, okay? This is not the structure of cell wall. I see. See, I get it. How do I say? Yeah, I will write a comment and tell people that they should watch till the end for the confusing facts, just because the start is, yeah, it's just because the start is not my style. Anyway, that was it, and bye.